Hello everyone, my name is Sophia Krombholz from the German Sports University Cologne, and I am happy to present you my virtual poster on the elimination profile of orally administered phenylethylamine. So, a quick introduction. Phenylethylamine, PEA in short, is an endogenous neuromodulator in the central nervous system, and by reuptake inhibition of noradrenaline, serotonin and dopamine, acts as a stimulant and is thereby prohibited in competition by WADA regulations. Urinary levels of PEA are influenced by various factors, including sport, and the question here is how to differentiate between endogenous PEA and that resulting from an application of the drug. So, in a pilot study 2015, a sulfo-conjugated metabolite, 2,3-hydroxyphenyl acetamide-sulfate, which I will just call M1, was identified, and that actually seemed characteristic for the oral administration of PEA. So the idea came up to use the M1 PEA ratio as a marker indicating the oral application of PEA. So the aims of this project were for once the synthesis of M1 reference material, the carryout of 2 elimination studies, first a single dose, then a multiple dose application, and at last the analysis of blank urine samples to suggest a cutoff level for the M1 PEA ratio. The study was designed like this. For the single dose, 100 mg of PEA were administered by 7 male and 7 female volunteers, and urine samples were collected up to 72 hours following application. For the multiple dose studies, 5 doses of 100 mg of PEA were administered on 5 consecutive days. First, only the morning urines were collected, then again up to 72 hours after the last dose. The urine samples were then analyzed by means of LC-HRMS. For the sample prep, urine was simply diluted with equivalent amounts of water and fortified with internal standard. Then separation followed on a new HPLC system with an end-capped C18 column using ammonium acetate buffer as eluent A and acetylnitrilyl formic acid as eluent B. Mass spectral analysis was performed on the Orbiter Explorer system by Thermo Scientific using both positive and negative ionization in full scan and targeted MS mode. So let's have a look at the results of the single dose study first. As we can see here, PEA itself doesn't really show an increase after application of the drug. We can see quite high natural fluctuations, which show that urinary concentrations of PEA itself are not sufficient to evaluate potential misuse. The sulfur metabolite, on the other hand, shows a clear increase within 12 hours post-administration, and maximum levels range between 29 and 272 nanogram per milliliter. So we see relatively high inter-individual variance here. And this also made it difficult to use the M1 PEA ratio as a marker, since the values were just not comparable between individuals and a corresponding cutoff level could therefore not be established. So instead, the administration study urine samples were searched for further metabolites that could reflect PEA intake, and a very considerable increase could be observed for phenylacetylglutamine, so PAG in short, which is highly concentrated in urine and had, as you can see here, a very similar course as M1. As already expected by the rapid turnover of PEA, the application of multiple doses did not show any signs of accumulation here. As a reference population, a total number of 369 urine samples of athletes were analyzed, and we can see here the density plots of the logarithmic urinary concentrations, and the colored spots each demonstrate the maximum levels observed in the administration study group. So, while the maximum levels of PEA measured in the administration study are clearly below the mean of the reference population, the urinary concentration of both metabolites are in general substantially higher after administration of PEA. Still, we could also observe a relatively wide scattering of the values, which makes it difficult to simply specify cutoff levels here. For this reason, multivariate binary logistic regression analysis was performed using the urinary concentrations of both M1 and PAG as variables to predict the probability of an oral administration of PA. If we now apply this model, we get a mean predicted probability of 1 for the urine specimen collected after administration of PEA and 0 for the samples of the reference population. So, with the p-values shown here, increased concentrations of both metabolites correlate significantly with the application of PEA, and we can see that logistic regression presents a suitable and new approach to detect suspicious samples in doping control. So in conclusion, threshold levels of phenylethylamine itself in doping control samples are not really adequate, but assessing both M1 and PAG combined can help to identify PEA misuse. For the future, IRMS could be a potential confirmation method to detect exogenous PEA, which would for once help athletes from facing wrongful suspicions and also help doping control laboratories to clarify suspect observations concerning PEA. At last, I would like to thank both my colleagues from the Manfred Donnicke Institute as well as the WADA for supporting this project, and Dimitra and Teresa from the University of Glasgow for the help and guide on statistics. And of course, thank you all for listening to my talk.